Good afternoon, all. I am Abhirupa Ghosh. I am a postdoc at Department of Biomedical Informatics, University of Colorado, and Shoots Medical Campus. I work with Dr. Jayani Ravi. So today I am presenting on our framework to predict antimicrobial resistance in escape pathogens using genomics and machine learning. So, as many of us know, that antimicrobial resistance or AMR has become a critical global health concern, and WHO has classified several bacteria as priority pathogens due to their high resistance levels and associated deaths. Among this, escape, a group of pathogens, are particularly notorious and are responsible of majority of health concerns and are often resistant to multiple antibiotic resistance. So the so traditionally the antimicrobial susceptibility testing uses culture-based method which are pretty slow to give results. Alternatively, there are molecular-based kits which are faster, but they are very specific and are restricted to few resistances. What we need are fast, high throughput, untargeted approaches, I believe, which can be achieved using genomics and machine learning. So, as we are starting on this project, we conducted a comprehensive literature review of current machine learning and deep learning based approaches for predicting AMR. So we wrote this mini review, and it is currently in press at Trends in Microbiology. We observed that most classical machine learning methods use genomic features to predict antimicrobial resistance phenotypes, and they also show quite promising performance and outcome features which are related to antimicrobial resistances. So, building on those foundations, we identified key opportunities and we tried advancing the AMR prediction methods. So, we are leveraging thousands of publicly available pathogenic genomes with AMR phenotypes attached to them, and we are exploring more than one molecular scale, such as including genes, proteins, and domains for deeper biological insights. We are also creating models that will support multiple bugs, multiple drug prediction, and we will integrate spatiotemporal metadata such as from where the sample was collected, the geographical location, the collection time or the year, or the host from which the sample was collected. This will give us a track of resistance evolution in different scenarios. Finally, we will be developing a scalable tool known as AMR for broad application across different pathogens and different data sets. So to work on this, we built this on few key biological questions, which are like, can we detect both known and novel AMR genomic determinants? Can we detect which molecular scale will perform better for AMR prediction? Or are there any certain features which are specific to particular time period or geographic regions, or the features which can give us, like, if they are shared across species or are they shared across drug classes? So to answer this question, we started by collecting assembled genome of escape pathogens from public databases like NCBI and Bacterial and Viral Bioinformatics Resource Center, also known as BVBRC. We built species-level pangenomes using Panaru and created a gene presence absence matrix. This served as the input for machine learning models alongside the corresponding AMR phenotype labels, which were obtained from antimicrobial susceptibility testing data. In addition to gene, we also explored other molecular level, as I was saying. We annotated the proteins and domains using Interpro and CD-HIT. This allowed us to create genome versus different molecular feature matrix, giving us more functional relevant feature space. So the models were developed using tidy model framework in R. We applied classical machine learning algorithms like logistic regression and random forest, and the training was performed using five-fold cross-validation. We evaluated the model performances using common classical metrics. We extracted the feature importance scores to identify which feature contributed most to the predictions and annotated those features to get functional information using different databases such as different methods such as mol evolver or interpro. Interestingly, when we look across the ESKAPE and multiple drug classes, we observed that logistic regression performed very well, uh, even when the sample sizes were lower than 1,000 observations per drug. Next, we looked into the features, which here I am showing an example of top features from aminoglycoside and Staphylococcus aureus model. So when we looked into the feature importance for aminoglycoside resistance model in Staph aureus, we see a mix of known AMR genes and virulence associated genes or phage associated genes, and there is a group of uncharacterized or hypothetical genes. So further, we used mol evolver to annotate those unknown or undiscovered gene, genes to get domains, and we found very interesting domains such as abortive. Okay, the second one, abortive infection, C terminus, or actin-like protein domains, which suggests that there is a potential of finding novel resistance mechanism or accessory pathways associated with resistance. We then shifted our focus to domain level models, and we see that when we are using the domains as annotated domains as features rather than genes, we captured more biological signals, and even we found the domains which were identified from the hypothetical genes in mol evolver. Next, we tried looking into the cross resistance among one drug class. So here I'm showing the example of aminoglycoside. So we explored the cross resistance among the drugs in aminoglycoside, which are gentamicin, kanamycin, or tobramycin. And we found that the gentamicin was able to predict on kanamycin and tobramycin data, but the opposite didn't perform very well. So when I looked into the top features, there were only few overlapping top features for the three of the drugs. And interestingly, the three genes which we found to be overlapped were part of other completely different beta-lactam drug resistance. This also indicates their possibility of multi-drug resistance evolution happening. Next, we went beyond drug-specific models and we examined different metadata-driven stratifications to find features if they are specific to certain sample groups or not. So here I am continuing with the example of gentamicin, where I went through building two country-specific models, one for UK, another for Singapore. And it is very evident to see that the UK model was able to perform well while identifying resistance in Singapore model, but the vice versa was not true. So the upset plot next, here also it shows that there were lot of individual features for each of the models, and this suggests that geographical stratification is important while developing AMR based models, and because AMR mechanisms can vary from one region to the other. Further, when we, so now, till now I was talking about one species, but gentamicin is one of the drug which is found resistant in across all the ESKAPE bacteria. So we compared the drug resistance model across ESKAPE to look how resistant determinants vary across the species for gentamicin. So interestingly, gentamicin, the top features for Klebsiella against gentamicin resistance were very different from another gram-negative bacteria, that is Acinetobacter baumannii. But when we looked into Staphylococcus aureus, which is a gram-positive, there were few similarities between the Klebsiella and staph top features. This also points to the possibility that there is a shared evolutionary mechanism of resistance across taxonomic boundaries. So to summarize our framework, the framework shows promising results from classical ML, which leverage genomic data to predict AMR across ESKAPE. These models capture known ARGs and implicate the presence of horizontal gene transfer and novel AMR mechanisms. It also shows that shared and unique features mediate resistance across drug classes and even species which are not related. The model's performance in gentamicin showed that it was greatly affected by geographical holdouts. So it would be it's very important to care about the metadata like geography or time frame or the host. And the resistance patterns, as we have seen, the resistance pattern vary globally, and comprehensive sampling or shuffling is also very critical to have for these models. So since this is an ongoing work, we are still working on improving our models. We are implementing different reshuffling methods and other classical ML methods. And we are also developing we are trying to answer more biological questions like monoresistance versus multi-drug resistance. And we are working on the package. We are working on the different data sets and documentation of the package. Thank you for your attention, and I would like to acknowledge my present AMR team and past AMR contributors, Dr. Jani Ravi, the PI and the whole lab, and NIARD for funding, and our department and computing facility for HPC. Thank you.
Great talk. I had a question on the UK and Singapore samples. Were they all from like one same collection center or one part of each of the country, or were they scattered, like, I guess, across?
So the UK sample was scattered across from different collection centers. But the Singapore, I guess it was just 2 of the collection centers. Yeah, it was pretty homogeneous sample.
Thank you.
Yeah.
Oh, that's fine. Just kidding. I was just wondering if you had looked at, in addition to gene presence absence, if you had looked for kind of like, SNPs that seemed like to kind of like, in conserved areas or something like that.
Yeah. So for the presentation sake, I was showing only gene presence absence, but we have 7 different data sets,
cool,
which also include k-mers.
Nice.
Yeah.
Nice. Cool. Awesome. Thank you.
Thank you.